Welcome back, everybody. It looks like people are using the discussion forum, which is great. We'll see how many of your questions we can get to in this hour. We'll have another opportunity tomorrow to talk. So I'm going to ask the first set of questions to there's a lot of questions about the exRNA sequencing pipeline. So I'll direct them primarily to Joel Rosowski from Yale, but please, other speakers, contribute if you have a slightly different take on the answer to some question. And if you want to ask a question live, please do that raise your hand thing at the reactions. And if we have time, I may open it up a little more than that. So Joel, the first question is, does the order of annotation have to follow genome, mirror-based, tRNA, PyR-based, lincRNA, and then circRNA?
So no, maybe I wasn't completely clear. So we have a default order of the annotations that we trust in terms of sort of confidence. But when one runs and that's the default for all the data that's been processed in the exRNA atlas, that's the order that we choose. But when one runs it yourself, you can customize the order. So if you've got a data set that's specifically targeting tRNAs, for example, you can prioritize the tRNA annotation over other ones.
Follow-up is, can whole genome come last? My thought was that it did come last. And
we simultaneously align against the genome, but the alignment of the genome doesn't affect the quantitation of the annotated RNAs.
What about RFAM and RNA central? I think I guess they're referring to different annotation suites. Joe, or maybe Juan Pablo?
Sorry, what was
the question was, what about RFAM and RNA central as annotations?
Yeah, what's the question? Sorry.
I think including them. I mean, we originally looked at including RFAM, but ended up going with the reduced set. I think it is possible to build your own database for excerpts. It's a little bit of a pain, but it's possible. So if you wanted to include RFAM and RNA central, which are perfectly fine databases, you could include those too. Or you could just take the genome alignments and do an intersect on those from the BAM file.
I mean, also just to add to what Rob said, that's correct. But I mean, the default annotations that we chose tend to be the ones that are highest confidence, and also the ones that tend to be the most abundant in the typical data sets that we've seen up to now.
Continuing on in the annotation frame for pyrna, which would you recommend, PyBase or PyRBank?
I think I dislike them all equally.
Yeah, one issue.
Excellent answer.
I guess one issue that maybe this is a question that I saw it was in one of the chats sort of a little earlier, is most of the annotations don't overlap, but we found, and Rob found, was that many pyrna annotations overlap, say, in known tRNAs, and the order which one implements the alignment makes a difference in that case, because it's like, if you've got something that overlaps, the order that you do it, either you can map it sort of simultaneously, or which annotation you prioritize. And I think that probably goes to Rob's confidence in the pyrna annotations. Is, I think some are better, but I don't think the pyrna annotations is definitely not up to the same level of quality as some of things like microRNAs in mirror-based, for example.
Completely agree. I think actually, sorry for being flippant earlier on, I think what Juan Pablo said earlier on is totally correct, that actually if you look at all the annotated sequences in PyRBase or PyRBank or whatever, most of them are probably fine. I mean, most of them are probably real. The trouble is, in a biofluid where you don't necessarily expect to see a huge number of pyrNAs, but you do tend to see a lot of ribosomal RNA, SNO RNA, tRNA, etc., then the ones that are run in the piRNA databases will probably be the ones that you will see in your exRNA sequencing. You know what I mean? So it's a little tricky, and that's why it's less trustworthy, I think, here than it would be in some other experimental context.
Along the same lines, I'll combine two questions. How does exRNA handle multi-mapped, non-unique reads within a single genome? And what about other RNA types? If you leave out the filtering of tRNA, for example, where do those reads go?
So, that's a very important question, and Rob will add. But the way that we do that, when we align against the genome, initially, one has a parameter in terms of how many places we allow things to map. I think it's less. I forget the exact default parameter. But the important thing is that that's why we line against databases of known annotations. So like microRNAs that potentially occur multiple times in the genome, we align against the database of known microRNAs. So the fact that a microRNA sequence occurs multiple times in the genome doesn't, reads don't penalize. And that's why when we sort of do the second pass alignment in terms of trying to identify things that are exogenous, then we allow a more lenient alignment in terms of things that can multi-map in the genome. So things that potentially align like 50 places or 100 places in the genome, we're not going to use in terms of quantitating known annotated RNAs, but we filter those out before we do the exogenous alignments. So we don't, so things that sort of repetitive sequence that are in the genome, we don't use for the exogenous alignment, but we don't necessarily trust them for doing the endogenous annotation quantifications.
I should have included this in that last question. Currently, exRNA output only tells you which parental tRNA a read comes from. Are you planning to implement more annotations for tRNA fragments like 5', 3' DERFs or internal fragments?
That's something we've been planning on doing, and at some stage we would like to implement that. Yeah.
You can get the exact positions of the alignment to the tRNAs again from the BAM files, but yeah, we currently just collapse it down to the anticodon just for simplicity's sake. But yeah.
And that's actually, Rob's correct, and that's actually true for all the longer transcripts. So if you look at like the read align to longer link RNAs or mRNAs or anything like that, you can actually, in addition to the quantifications, you can go look at the BAM files and actually look at the actual alignments. But we don't quantify discrete sort of fragments of longer transcripts.
Correct me if I'm wrong, but we'll discuss this more tomorrow in a session on looking at exRNAs with RNA binding proteins, but you can tweak the way you filter annotations when you're doing an exRNA run. But as Rob and Jill have both said, the output of the output that exRNA creates, like on the exRNA atlas that crunches through everything, there's a BAM file with read pileups that you can then look for your own custom annotation. And yeah, it's tweaky a little bit because you've piled them up previously, but it's a good first pass without having to rerun the whole analysis. Can a custom database be used for exRNA as an endogenous source instead of HD38, HD19, MM10?
So Rob alluded to that. Yes, if one goes and installs the code yourself, one can sort of replace the annotation files with custom annotation files. If people are interested in doing that and need some help, feel free to sort of email me and we could sort of potentially help. People have done it. It's not, I mean, it's a little bit tricky, but it's doable.
I think it gets a little fruity if you have to add biotypes that are not in the kind of priority list already, because you'll then have to go back to the Java engine and recompile that too.
But if it's like just substituting a newer set of trna annotation, that's
absolutely
that's not that hard.
Yeah, yeah.
I know that Peter said he had to go, but I think he might still be here. Maybe not. I'll ask this question anyway because others could answer. Can we perform differential expression analysis across different biofluid samples without skewing because of the differences in library size?
What do you mean by library size in terms of the number of reads or the way the experiments were done in the different biofluids?
My interpretation is that they're talking about number of reads and asking about normalization. That's the way I interpret this question.
I mean, you probably could. I would not sort of recommend comparing healthy urine versus cancer plasma. You know, that doesn't make sense to me, given the huge difference in the RNA content of the biofluids. And so if you're asking, can you run some kind of linear model with two levels with, you know, plasma and urine and cancer and healthy, one in each biofluid? Well, technically, yes, you could, but I think I would do those separately. DEC2 does normalize for library size. That's correct. Somebody wrote, I mean, all these differential expression tools normalize for library size. I think the question would be the vastly different complexity of each of the biofluids. And I would probably recommend you just run them in pairs per biofluid. So do your DEX in plasma separately to your differential expression in urine. I think that's probably safer, and then see what's consistent or not between the two. I don't know if, unless you're looking for an interaction term between biofluid and disease status, I'm not sure it is beneficial to run them at the same time in the same model.
So before we move on to other classes of questions, I'm going to unmute everybody. If you have a question, please unmute yourself, ask it, and then mute yourself again. There's nothing worse in a discussion than, you know, hearing somebody typing or telling somebody in the lab to order, you know, pipettes. And so we'll see how it goes. If you have questions on this specific, basically the RNA-seq processing pipelines, now's the time.
Sorry, yes, this is Angie from OHSU. So I was the one asking regarding if there is any way that we can perform the differential expression analysis when you have totally different library sizes. Like you mentioned, maybe across different biofluid, not necessarily between cancer and healthy.
Okay, did any of the answer that I suggested make any sense, or do you still have questions?
I think because now we are analyzing more larger set of different types of biofluids, I don't think there is actual established methods to analyze this differential expression analysis. So I was wanting to know if someone has an idea about this approach.
One thing to just, one other idea would be to use some spike in
Yes.
that you at least can normalize to your input in that way. It's another thought.
Yeah, but if we use the ERCC or other spike in, considering there's a huge library size differences, we might still have a skewness to the overall expression being changed.
But sorry, when you say library size, do you just mean sequencing depth?
Yes, the total inputs of your samples.
Yeah, I mean, like people have noted, I mean, most of the differential expression that people do in biofluids is similar, if not identical to what you would do in tissues and cells, which is fine. I mean, there's a number of different tools out there that will do it for you. Somebody in the chat mentioned, Andrew mentioned DEC2, which is one that's extremely liberal and makes a lot of assumptions that sometimes give you spurious false positives. I think, you know, there's the Limavoom and Edjar are ones that are a little bit more conservative sometimes and maybe a little bit closer to reality, I think. But again, they all make assumptions over library size. They all do some form of filtering on the low end of expression. I think my worry with the very different biofluids would be if you had, a very simple biofluid, maybe like CSF, where you don't, I mean, it's very complicated in reality, but when you end up doing the sequencing, you see maybe a smaller amount of RNA in total, and you see a more sporadic set of RNAs expressed, that you would be quite often comparing something versus nothing in the two different biofluids, which may present challenges for normalization and filtering. And if you have certain very high, like in plasma, if you're looking at mRNA in plasma, you get an absolute ton of globins, right? You get a lot of platelet RNA, you get a lot of HBB, HBA, the hemoglobin genes, complement, all sorts of stuff floating around in plasma that simply doesn't exist in some of the other biofluids. And because of the way that sequencing works, you may end up saturating your runs with those reads that then suppresses everything else. And so I think if I was you, I would not wish probably to do the differential expression between biofluids and treat them separately and just look at them as being confirmatory, possibly.
But just to add, I think doing differential expression between different biofluids where everything else is is done the same. I mean, there are assumptions, but I think that's reasonable. I think the issue becomes more significant where the two different biofluids, where there's some parameters in terms of the way you did the experiments are different. So if you a different isolation kit or different library construction kit, anytime you introduce additional parameters beyond just the biofluid, I think then it becomes a really big issue, and issues of normalization. I don't think there's any standardized way of doing the differential expression.
Thank you for the answer.
Oh, sorry, I was muted. I asked if there are any other questions on this topic of exRNA or other RNA-seq processing pipelines, live questions before we move on. So the next sort of list of questions may be a bit scattershot in sort of jumping from topic to topic. And please keep in mind that some of these are pretty introductory questions, and maybe your answer will be a repetition of something somebody said in a talk, but that's fine because, we've got a lot of people new to the field and repetition is a good thing. So this is the $64,000 question. I'm hoping our speakers can comment on carriers of RNA. Are EVs really the main or most important carrier? I can take a stab at it if no one wants to jump into it. I would say that EVs are the best understood carrier, but by no means are they the only carrier. They're probably the best studied. We have a session tomorrow on exRNAs in connection with RNA binding proteins, which would be basically anything that can protect an RNA from degradation in the biofluid can be a carrier. So EVs hold them inside, RBPs bind them and protect them, different kinds of lipids, HDLs, LDLs. We're less far along in our understanding of exRNAs in other kind of carriers, just because I think the experiments are difficult. With RNA binding proteins, you need an antibody for each specific class of protein. But again, we'll talk about that a bit tomorrow. Does anybody else have something to add on that?
Hi, Roger. If I would like to jump in regarding, of course, I agree with you that you need an antibody to study a specific RNA binding protein, so the RNA associated to a specific RNA binding protein, like argonaute2, for example. But then you can just look at a broad view comparing the amount of RNA in ultracentrifugation supernatants versus the pellet, And we have done that and many others. And in cell culture, at least, most of the total RNA yields are higher in the supernatant. So this shows that there's quite a lot of RNA outside vesicles. And we heard some talks today about the fragmentation pattern of different mRNA fragments in plasma or serum that are compatible with RNA treatment, meaning that are compatible with the degradation by RNA family members, which are abundant in serum and biofluids. This shows that at least some of these mRNAs are accessible to ribonucleases. And we have done the opposite experiment, just adding ribonuclease inhibitor. And that shows that we can really shape the exRNA profiles. So I think there's quite a lot outside vesicles, but of course, vesicles are much more studied and there are more tools to study them.
Sort of related, I want to plug some of Juan Pablo's work. I think that our field has a sort of default mental paradigm when we think about this stuff, that there are RNAs that are inside the cell and they get secreted into a vesicle by some mechanism that we need to figure out. And then the vesicle goes and does something in a target cell. But Juan Pablo's done a lot of work showing that, you know, a lot of stuff gets secreted into circulation, and the ones that are the RNAs that are most stable against the degradation are what's left. And so it's not so much, in a lot of cases, it's not so much a secretion process as a sort of survival in the outside environment process. And so we need to keep both of those paradigms in mind when we're thinking about a particular piece of exRNA biology. So let's just jump around a bit. Rob, how does EV liquid biopsy overcome heterogeneity if you don't know where things were secreted from?
It's a great question. I think it's a very good question. I think, I mean, if you do some kind of, I mean, you know, we were talking about this before, but if you do some kind of biochemical enrichment using an antibody, I think that's one of the strongest potential, you know, causal's the wrong word, but strongest links that you have to a cell type of origin. I think Peter said this extremely eloquently, and it was something I forgot to mention in my talk, is that, you know, your deconvolution is only as good as the signature matrix that you're applying to it, right? Like, you need to understand, you know, ideally to do this well, you would need to have, you know, single cell resolution atlases of the entire human body, and then figure out, you know, exactly what the contributions of each of those cell types were to the biofluids, which I think is not actually that far away. I think that, you know, there's projects underway that will probably help fill in some of those gaps. But it's a great point. I think that the reason that we focus on exosomes and EVs so much is because they have this potential, Alex is about to correct me again, is that you know, they are actively secreted either via the multivesicular bodies and the endosome or from the plasma membrane. And so it is, you know, it may be that, you know, that there's some potential for a cell type enrichment there. The question, though, again, and I didn't mention this either, is that you're always assuming that the release of RNAs into these vesicles is stochastic, right? That it's some form of random profiling of the cytosol or the cell from which it's derived, which for each individual EV is not true, for sure, because they're so small that they only contain a very small number of RNAs. But we don't actually know that, and it's an assumption that underlies all these cell-based deconvolution methods, which is not what Alex was presenting. But you know, all these, if you're hoping to say this mix of tissues gives you this plasma exRNA profile, you have to assume that each of those tissues can contribute equally, which may mean that the RNAs in the cells are stochastically loaded, and also that each cell is equally likely to dump an EV into, say, the bloodstream, which may be different for different tissues based on vascularization and just proximity to the biofluid. You know, it's not reasonable to assume that your liver is going to be contributing EVs to the spinal fluid. it may be in very, very small amounts. I don't know, but it's probably not the norm. So yeah, there's a lot of assumptions that go into the cell type of origin, which sort of get bypassed a little if you can go in with antibodies and pull out a pure population of EVs that have, you know, some marker on them that's specific to a cell type. Then it makes your life much easier then to try and pick apart whether they're actually derived from that cell type or not, if that makes sense.
So Rob, I'm sorry to say this right after your eloquent response, but we have so many questions that I'm realizing now to get to. Speakers, please try to keep your responses brief and pithy from this point on.
Sorry.
You know, yeah. So this is probably a question for Juan Pablo. You know, he has multiple articles about this issue. Does the high amount of ribosomal RNA come from exosomes?
Well, that's a good question. Just to be brief, we have recently published a paper showing that there are also ribosomes outside vesicles, at least in cell culture, but there are also some insights that they might also be in biofluids, and that makes sense if a cell is broken, because if a cell dies, most of the RNA that will be passively released will be actually ribosomes, and they will be outside vesicles. So for seconds at least, for short amounts of time, they will be there. So I think that the non-vesicular fraction contributes a lot to ribosomal RNA. Now, are there also high quantities of ribosomal RNAs in vesicles? Are there ribosomes inside the biggest vesicles? I think these are still open questions.
Yeah, and actually, just very briefly, it comes back to the stochastic loading thing too, right? Like 18 to 95% of the RNA in each individual cell is ribosomal, probably. And so if there's a stochastic loading of that into EVs, then it makes sense that the EVs would be predominantly ribosomal RNA as well. I mean, they're in the protein complex of the ribosome, which is a little different, but you know, same deal.
Jumping around again, how do you plan to tackle, I guess this is for maybe Matt and Joel, how do you plan to tackle the biases of sample sources, for example, ethnicity, age, gender, in building and analyzing the atlas?
I'm not sure we actually have all that available metadata. I don't know if we have all the like ethnicity, but I think
we do.
We do.
Or if there is an option to include that. I believe it is mostly, almost everyone fills it out. So we do, yeah.
I mean, I think we're gonna, in terms of the analysis, we're gonna look to see whether we see any sort of significant differences. I think if we observe sort of differences, then we might have to sort of think more about sort of trying to use that in terms of normalization.
And also, too, is the Tawari lab. I don't remember if it got added to the ERCC work, but we had 100 healthy individuals, 50-50 gender mixes with ethnicity in it as well, and we really didn't see much of anything in terms of differences. But I mean, that was a fairly large data set, and you know, just that we did ourselves.
Yeah, so I think actually each of those responses kind of answers that in some ways. Just regarding the stage 2, the studies, you know, there's a fair number of samples, as I mentioned, that will be analyzed over the next couple of years, but those are really driven by the different investigators who submitted proposals and for whatever reason they selected certain samples for certain technologies, whatever. So, but it's a good question, but I think several aspects are captured. But anyone who has samples, regardless of their origin, we can capture that metadata.
Moving on, what about long non-coding RNAs? Are they found in vesicles, and how are they annotated?
In terms of full length, I mean, this goes, Rob, it might be best in terms of answering.
Yeah, I mean, we see long non-coding RNAs in vesicles, just like you see pretty much almost, I think, every RNA biotype in EV preps. and they're annotated in, I mean, just based on whatever long non-coding RNA annotation we have available. I think the default is to use the most recent version of GenCode Ensemble. But obviously there's other databases out there as well. I think they tend to be a bit more, they can be more cell type specific, which may help you actually in terms of deconvolution.
And in my experience, maybe the biggest contributor is Mallet1. You see most of that's sort of on the top of the list, and Meat1 is also on there too, but there's usually a few that sort of dominate those reads that I've seen.
Right, absolutely. And they tend to be the ones that are highly expressed in across cells as well. Yeah.
Ryan, will you put those two in the chat?
Sure.
I should have probably asked this in that last big chunk of questions, but for Ryan, how important is it to first map to UNIVEC, especially in samples with reduced exogenous contamination potential like seminal plasma?
I just do it as a default. I think it's very important to put the UNIVEC in there. I mean, that's just kind of default in what I do to get rid of those. You'll see a lot of like stop oligo and other things too that come through. You want to get those removed.
So basically that's a first step in pretty much anybody's pipeline. Can you see Ryan, can you see PCA3, which is used for diagnosis of prostate cancer using phospho-RNA-seq?
I would have to look. I don't remember all of them specifically, but if you look at our EMBO paper, so EMBO and Ryan Spangler, we have a list of all of the messenger RNAs and non-encoded RNAs that we detected.
And also for Ryan, can you differentiate genomic contamination from actual coding RNA reads?
Yes, and I think Joel may have talked about this as well, but we've mapped, we looked basically at the reads that were mapped in the same orientation as the transcript and compared that to the ratio that mapped the opposite orientation of the transcript. And we since it's a stranded protocol that we're using, and we see that at least after our filtering, we see an enrichment of exonic reads in the sense orientation, which makes us pretty confident that it's not genomic contamination.
Well, also you could look for gapped alignments rather than just things that map to the genomic alignments.
Good point.
This is for Klaas. Could a pregnancy test also indicating trimester be made based on the measure of microRNAs, and in which fluid would we obtain those microRNAs?
Based on microRNAs, I think there's a discrimination between trimester 1 and 3, or the later stages, trimester 1 from 2 and 3 possible. Possibly including other RNA classes, there may be a better discrimination possible. And the biofluids we used so far was actually the biofluid was plasma.
Thanks, Ryan, for putting those in the chat. This is, I guess, an early process question and a sort of experimental question. I'll ask Rob, but others can answer. What are your thoughts regarding protease and RNase treatment to eliminate contaminating RNAs outside of EVs?
I mean, I'm probably not the best person to ask, actually, having failed at being an experimental biologist. But I think it's been done to prove, especially with the question over whether cell-free DNA is contained within vesicles with nuclease digestions and things.
When I said Rob would be the best person to answer, I meant Juan Pablo. I apologize.
I think that should be your default, actually, Roger.
Well, of course, that depends whether you're definitely not interested in non-vesicular RNAs. But even if you want to focus on the RNA which is inside vesicles, it's like a standard procedure to show that they are protected by RNAs. And when you combine a detergent and then RNase treatment, then you make the RNAs accessible. But it's also regarding RNase treatment alone, it's good to also take into consideration that there are also ribonucleoproteins, which they are proteins which protect the RNAs against the action of RNase A. or which is found in serum. Otherwise, we wouldn't have RNAs outside the vesicles circulating in serum or plasma. So I wouldn't say that RNase-A treatment alone is a sufficient proof that we are working now with intravesicular RNA. A combination of treatments might be more informative, but there are always caveats.
If Justin is still on, maybe he could lead off answering this question and then be supplemented by others. How does deconvolution compare to clustering?
I haven't experimented much with deconvolution. I believe I attended a presentation by Rob, which showed some pretty promising results from the same data set. So I think it's a good idea, and it could be implemented as another dimensionality reduction if it continues to be, if it's useful and pre-computable. Yeah. Also, I believe the deconvolution usually uses the labels. I could be mistaken, but at least in the presentation, it might use the label. So I guess it would be supervised versus unsupervised, but I could be wrong on that.
I'll note again, we have a whole session on deconvolution tomorrow, but does anybody else have something to add there?
Well, deconvolution doesn't really use labeling. The common feature that enables both clustering and deconvolution is non-randomness in data, right? The fact that you can explain a lot of variance using small number of, say, principal components generally suggests, that there is a chance that you may deconvolute the matrix, into two matrices of low rank. So they benefit from a similar type of pattern in the data, but they're very different, methods. So, mathematically speaking, reference-free deconvolution, right? It's not the kind that Rob Kitchen talks about. You can think of reference-free and reference-based deconvolution. So what we've seen today is just reference-based deconvolution, where you know what the components are, and you do something like multiple regression, really, not deconvolution. That one is reference-based. There is also reference-free deconvolution, like exDEC that was used for the exRNA atlas that I'll talk about tomorrow. That is essentially matrix factorization. So principal component analysis also is matrix factorization. It's just that the type of factorization is different. Essentially, you have a large matrix and you represent it as a product, of two matrices. So both principal component analysis and reference-free deconvolution fall into the same category of problem. That was a long answer.
Alex, I appreciate that. I don't think we have time to go further into it right now, and you'll discuss it tomorrow. But I think this issue of, a gold standard of reference data set for finding tissue of origin or exRNA carrier versus your reference-free methods, I think that's a key point for people to understand. So I hope we do understand that better tomorrow. And while we're on the topic of deconvolution, let me ask you and others two questions. Can deconvolution be used to get cancer tissue of origin for liquid biopsy diagnostics? And an earlier question for Rob, why do you think such a test would never be approved for clinical use?
Who wants to go first?
Anyone?
I could say, I'll be talking a little bit about it tomorrow, in my session. exDEC method that we use for exRNA also is used for tumors, and our program is exactly that, to link heterotypic interaction within tumor tissue with liquid biopsy biomarkers. And actually, we pulled a thread on glioblastoma in a paper published earlier last year, where we shown that, using deconvolution, we found difference in glioma and glioblastoma in terms of cell type composition identifying miR9 as a microRNA where cancer cell-induced angiogenesis, and that miR9 is also elevated in the plasma of glioblastoma patients, but not glioma patients. So we kind of pulled thread on it, and the key element of that strategy is actually to use the same deconvolution method for both the tissue and liquid biopsy.
And Rob, why will this never see the clinic?
Well, no, I don't think I said never. I think it would be difficult, and I think it's partly the regulatory environment is much more used to very small numbers of analytes. Your PCA3 is a good example of that, right? It's like a single a small number of markers that distinguish prostate cancer or aggressive prostate cancer from benign. The other part of it is just cost. I mean, to do the kinds of deconvolution that I'm talking about, where you have a reference, where you're hoping to try and understand relative fractional contributions of various things that you quote unquote know about to a biofluid, you pretty much always have to measure the same stuff by the same technology. And if you're talking sequencing, makes it a very expensive test. So if you're hoping to bring a diagnostic to the clinic cheaply, in a way that is understood by the current regulatory authorities in the States, at least, PCR is much easier, or a protein-based assay is much easier to do that.
It's clearly not the topic of this workshop, but I think for the field, this issue of how we integrate exRNA-based biomarkers into the clinical milieu of existing protein, maybe circulating tumor DNA biomarkers, other kinds of diagnostic tools, I think is key. And it reminds me that I should plug an online class that we're developing about extracellular RNA. One of the lectures of which we'll discuss that very topic of how to integrate these new biomarkers into clinical practice. Stay tuned, maybe that will come out in the fall, hopefully. So here's a question, and I want to piggyback on top of it. And mine is a stupid question, and I feel fine asking it. So the question is, why must we focus on blood, plasma / serum when we do sample isolation? Can't we be missing important RNAs that are not secreted in blood? I think the answer to that is we can do lots of different biofluids. But my question on top of that is, why serum and plasma rather than whole blood? When you're looking at whole blood, what extra are you looking at that you're not finding in serum and plasma? I wish Kendall were on. I think she can answer.
I mean, my take on that is that you just end up getting so much more from red blood cells and platelets that you won't see the smaller signal from organs that you're probably interested in. If you want to look at whole blood, you probably just look at the immune cells or the red blood cells and white blood cells themselves rather than cell-free populations.
Completely.
I might actually approach you offline about this question, Ryan, because I have a couple of research projects where they look at whole blood and I don't understand why. Maybe you can be able to explain it to me.
I can support Ryan's view. We've tried something similar and it's really devastating. You pay for millions and millions, hundreds of millions of read depth, and all you end up with is transcripts from blood cells ultimately. So it's we were hoping to capture something that might have been lost due to the processing of plasma or so. And I mean, for sequencing, this approach without any kind of enrichment is a total failure, I think.
I think sometimes whole blood can be easier to handle potentially. I think there are sometimes advantages in storage and in transportation of whole blood maybe, but I agree completely. I think even when you're looking at plasma and serum, the majority RNA contribution is still from cells of the blood, not necessarily just red blood cells, but platelets and white blood cells. And so, given that's the case, then, if you add the cells themselves, it's only going to get harder to find a tissue-specific signal.
And to answer the other person's question a little bit, drawing blood is one of the main things people do, you know, when they're diagnosing disease. It's sort of, but as we've seen, you can look at saliva. There are other biofluids like cerebrospinal fluid that are harder to get to. Basically, whatever is easier than doing the clinical standard of an invasive biopsy is what our field is interested in. And on that point,
Yeah, Roger?
Carolina might be.
Oh, sorry, go ahead.
Yeah. No, go ahead.
I think you kind of nailed it too. It's a simplicity, a widely used assay, accessing blood. And I think the complexity issue is what people mentioned. I think it's, I don't know, 8 or 10 logs of complexity in terms of quantity, different transcripts and different particles, et cetera, in the plasma. But regarding the profiling of other biofluids, in stage 1, there were about a dozen different biofluids. Again, a large focus on plasma or more readily accessible biofluids, but there are a number of other ones which, again, you mentioned CSF or urine, which are commonly assayed for various, disease states, et cetera. So there's a breadth of different biofluids, even though the focus is on a few.
So Matt, are you saying that one of the reasons people focus on blood products is there's a richer source of RNA, there's more data in it? I know CSF, for example, is very sterile, and so maybe there's less.
And I think what you were saying, it's widely used. Everyone, you can draw blood, right? But then the question about, other people comment on the complexity of plasma and serum. So if you got to go into fractionation and et cetera, what you do need to do if you want to get to low abundant species, which is part, again, stage 2 is how to get to these, low abundant species, whether it's particles or cargo.
And on this topic, and maybe Carolina might have some insight. I don't know if Minu is here. Anyway, in order to make a biomarker detection system in urine.
I am here, but I didn't hear exactly the question.
Oh, I'm reading the question right now. In order to make a biomarker detection system in urine, we would also have to take into account exogenous RNA, and how could we make sure we're measuring RNA from tissues in that kind of setup?
This would be the urine samples, yes, and for the biomarker. I think like that it could be similar approach like to the saliva, like to check, for example, what would be the alignment for the if it's from the, for example, if we would be interested in the human biomarkers, I would maybe do two pipelines to compare, like the direct alignment to the human genome. And then the second approach, I would, for example, I would firstly do the alignment to check for the exogenous reads, like, for example, bacterial. But I don't know in urine what we should expect the most. I don't expect that in urine we would have a lot of bacteria. I don't think so. So I think
I don't mean to put her on the spot, but I wonder if Minu has any thoughts on that.
And then
she may not be available.
Actually, regarding this question, we've analyzed urine and we had sometimes 90% bacterial reads when we characterized small RNAs, and that was relatively directed. So I wonder if you look at mRNA fragments or so, it would be even worse. I would suppose so, since this environment is very highly it has a lot of ribonuclease activity. They would be highly fragmented. I think this is extremely difficult in urine. But of course, if there were transcripts, discrete transcripts, and I think from the tissues, you sometimes, from the tissues of the urogenital system, you do sometimes find transcripts, even full length in not sure in which environment, but we've observed a few transcripts, and that turned out to be a biomarker, probably a method that is directed at those specific transcripts might work.
It's worth also just quickly noting that the first commercially available diagnostic test for prostate cancer, in fact, of any disease with exosomes, a male company was a urine test for prostate cancer, and you can see PCA3 mRNA in the urine. You can see a couple of other markers as well that they use. So I think, you know, there is potential for exogenous signal that might also be diagnostic in some cases. If you're thinking about an infection of the bladder or something, then you might be able to detect it. It may not be bad that you can detect exogenous sequences. But for sure, there's a bunch of human mRNA in urine exosomes or in the urine. And this is in the NCCN. I mean, this is a now a commercially available test that you can get if you have a gray zone PSA score for prostate cancer.
I think we'll leave it there since we're at our 50-minute mark. There are some unanswered questions. What I'm going to do is I will collect them, and then I will clear again this discussion forum 700500 on openquestions.org. And if you feel that a question you wanted to get asked didn't, and you really would like an answer, just re-upload it to there, and I'll make sure that we get it asked tomorrow. So again, tomorrow we have a session on deconvolution. And trying to remember what is happening tomorrow. Hold on. Session on deconvolution, and then that session on exRNAs connected to RNA binding proteins rather than vesicles. 3 case studies of specific analyses, and then another hour of discussion, and then Gustavo Stolovitsky, who co-founded the DREAM Challenges, is going to lead a discussion about what kind of exRNA data analysis challenge we might want to make as a community. So thanks everybody, and see you tomorrow.